Welcome to today's lecture on embryology. Today, we will be discussing the development of the ovaries. We will cover the stages of ovarian development, including primordial germ cell migration, follicular growth, and hormonal regulation. Get ready to dive into the fascinating world of the female reproductive system. Let's begin. In a video about the development of the ovaries, we explore the migration and differentiation of primordial germ cells into oogonia, the precursor cells for eggs. We observe their journey from the endoderm and mesoderm, guided by chemotactic cues, towards the urogenital ridge. Upon reaching the ridge, the oogonia undergo distinct developmental stages, including proliferation and eventual meiotic arrest, which ensures no further division until puberty. In the early stages of ovarian development, the urogenital ridge forms from the condensation of mesenchyme. This structure provides the foundation for further gonadal development. The gonadal cords, which consist of primitive sex cords, originate from the urogenital ridge. These cords will later differentiate into granulosa cells that surround and nurture developing oocytes. The early gonadal cords serve as the groundwork for follicular development. In early development, primary oocytes undergo mitotic arrest, stopping their proliferation. This is crucial for future ovarian reserve, preserving oocytes for reproduction later. The mechanism involves an activation of CDK by Airb, leading to the cessation of oocyte division. In the fourth section of the video, we will explore the development of primordial follicles and their role in determining the ovarian reserve. Primordial follicles consist of an immature oocyte surrounded by flat granulosa cells. This crucial process occurs during fetal life and establishes the finite number of oocytes in a female's lifetime. In the fifth section of our video on development of the ovaries, we explore the complex process of follicular activation and hormonal regulation. We focus on the role of follicle-stimulating hormone, FSH, which stimulates the growth and development of primary follicles through the proliferation and differentiation of granulosa cells. Paracrine factors like bone morphogenetic proteins, BMPs, and growth factors also contribute to follicular growth and the delicate balance between recruitment and atresia. As follicles mature, the squamous granulosa cells that surround the oocyte undergo a transformation into cuboidal/columnar cells. This transformation is essential for the production of essential follicular components and contributes to the growth and development of the oocyte. Follicular proliferation occurs simultaneously and is driven by the proliferation of granulosa cells, ultimately leading to the formation of a mature, preovulatory follicle. In the seventh section of our video on ovarian development, we explore the crucial process of theca cell differentiation in the follicular wall and its significance in androgen production. Theca cells, derived from stromal or existing granulosa cells, form distinct layers known as theca interna and theca externa. Theca interna cells produce androgens, such as testosterone, stimulated by luteinizing hormone, LH. These androgens are then converted to estrogen by granulosa cells, aiding communication within the follicle. Antral follicles represent the advanced stage of follicular development and are characterized by the development of a fluid-filled cavity called the antrum. The formation of the antrum involves the accumulation of follicular fluid, secreted by the granulosa cells, which creates a dynamic environment within the growing follicle. This fluid-filled cavity not only supports the oocyte but also aids in the process of follicular selection and dominance, determining which follicle will eventually ovulate. Ovulation is the highly orchestrated process by which a mature oocyte is released from the dominant follicle to facilitate fertilization. This event is triggered by a surge of luteinizing hormone, LH, which occurs as a result of positive feedback between rising estrogen levels and the hypothalamic pituitary gonadal axis. The surge of LH induces a series of biochemical and morphological changes within the follicle, leading to its rupture and subsequent release of the oocyte. Following ovulation, the remaining follicle transforms into a specialized endocrine structure called the corpus luteum. The corpus luteum is responsible for secreting progesterone, a hormone essential for preparing the uterus for a potential pregnancy. The formation of the corpus luteum is characterized by the luteinization of granulosa and theca cells, leading to the activation of numerous biochemical pathways that facilitate progesterone synthesis. In the absence of fertilization, the corpus luteum will eventually undergo regression. This process involves the atrophy and degeneration of the glandular and vascular components of the corpus luteum, leading to its transformation into the corpus albicans. The corpus albicans is a remnant structure in the ovary composed of fibrous tissue, marking the end of the reproductive cycle and preparing the ovary for future follicular development. Hormonal signaling pathways, involving FSH, LH, estrogen, progesterone, and inhibin, regulate ovarian development. Key players include the hypothalamus, pituitary gland, and ovaries. Understanding these signals is vital to grasp ovarian development and function. In conclusion, studying the development of ovaries is crucial for understanding the female reproductive system. It involves complex interactions between cells and hormones, from germ cell migration to follicle formation, growth, ovulation, and regression. This knowledge helps unravel embryology and reproductive function.